All right, so my name is Tory Blackwell. Again, I'm an instructor over at Clackamas Community College. And today I'm going to be... Today I'm going to be talking to you about elevator pitches. Now, it's early, and sometimes brains need to warm up. So on your tables, you should all have an orange and a banana. This is actually a pretty fun activity. One of the things that, that you need for an elevator pitch is kind of a question. So, we're going to use that orange and that banana to help us kind of think about asking questions, and hopefully that puts people in a mindset that'll help them with developing their elevator pitches. So what I want you to do is, as a table, grab the orange and the banana and take maybe 5 minutes to see if there are a couple of interesting questions that you can ask about that orange or about that banana. Like, I will give you one, but then you can't use it. What makes this orange orange? Like, that type of a question. Something that you might be able to study. Right? Take about 5 minutes, and during that 5 minutes, you're also going to be getting some handouts related to the elevator pitch. So now what I want each table to do is you're going to share your question with the rest of us. It's a good chance to do a little bit of public speaking because there will be more public speaking. It's a thing. So I'm just going to start over here with this table. Just one person at each table. So, pick one person at your table to share your question. And it can be about the banana or the orange. Oh, and if you can also say your name and where you're from before your question so that we can start to put more faces and names together.
I'm Monica from PSU. One of our questions was where the bananas originate.
Okay, where bananas originate. Let's go over here.
My name is Mario from PSU. One of our questions was, who named it banana?
Okay. Bananas get their name, alright?
My name is Amelia and I'm from PSU, and one of our questions was what is the lethal dose of bananas?
Okay, that can be an interesting ethical dilemma, alright?
I'm Max.
Okay, oh, one second, okay. Calm down. Alright, go ahead.
I'm Max, and I'm from Alaska, and we had a couple of questions about the orange. One is, why is it green on the bottom? And another is, why does the skin have dimples?
Okay.
Hello. So we wanted to know why the orange already is like kind of in slices. And that was our question.
And I forgot, where are you from?
Oh, sorry, I'm
okay.
from PCC, PSU,
Okay.
kind of.
Hi, my name is Mia. I'm from PSU, and our question is, which came first, the color orange or the fruit?
Okay.
I'm Oliver from PSU, and we have a couple questions on the banana. The first one was, why can't bananas grow individually? And the second one is, what do you call multiple bananas?
Remember, this is a safe space to explore and ask questions. Okay?
I'm given orange with the wrong group, and our question was, why is the banana dish really original shape?
Okay. How does it develop its shape? All right, so it looks like we're hitting this side. Sure.
My name is Stephanie from Clackamas Community College, and our question is about the orange. How does the acidity of the orange affect the tree's evolutionary fitness? How did it, if at all?
Our question was like, how many gallons of gas did it take to get the banana from where it originated to here?
I suspect you don't want the answer to that question.
My name is Isabella. I'm from PSU, and our question was about the banana and like why it's naturally split into thirds.
My name's Conrad.
Okay, hold on.
My question is, you say this is a banana in orange, but I doubt that that's an orange. Is it really an orange?
Ding, 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 ding. It's a mandarin.
Hi, I'm Amber from Chemeketa. Our question was, what is the chemical makeup of the orange and the banana?
Okay.
I'm Crystal from PCC, and we had two related questions. One, why do they both have peels, and is that related to some type of defense mechanism for the fruit and the plants as a whole?
I think that leaves our last table. Well, not those two. They can't play.
I'm Adam from PCC, and we were wondering why banana becomes spotty when it starts to become overripened. Why is it more like consistent, like turn black, white, forms like little dots?
Okay. All right, so thank you for sharing all of those great questions. Some things that you might have noticed if you were sort of paying attention. Some of them were very botany. Some of them were almost like linguistic, anthropology, more social sciences. Some of them were chemistry. Some of them were medical, you know, when you're thinking about toxicology and things like that. Right, there's a lot of different ways that people can approach their questions, right? And a lot of times those questions, like the way that they approach them, are probably based in what their interests are. Or it could have been based in, it's 9 o'clock in the morning and I need to come up with something fast. There's a little bit of both. So I want you to kind of keep that in mind while you're developing your elevator pitch, right? Like what was it that kind of got your noodle going, right? So that you can use that to help you and get into that right mindset when you're trying to develop your elevator pitch. So, first big question, what is an elevator pitch? We've got this nice list of statements on the board. A short statement of your research interests and experiences. Okay, that seems sort of reasonable. How do you share it? Informally and orally in various professional contexts. Professional contexts, here's a list of 4 different contexts. How many of you walked into the room on, what day is it, Thursday? On Wednesday, absolutely nervous, like to the point of shaking, because you're going to walk into this room with a whole bunch of people at a conference and you know none of them maybe. Anybody? It's okay. This is a normal thing, right? But imagine if you had the ultimate useful icebreaker, right? It's great to have some type of icebreaker so that you know you have something that you can talk about when you get into these settings. And that's what an elevator pitch becomes in a lot of respects, right? So what's an elevator pitch sound like? So again, I'm Tory Blackwell, and as a graduate student at the University of Illinois, Chicago, I spent most of my time looking at yeast. I was specifically interested in yeast signal transduction pathways, how individual yeast cells can communicate with each other. It turns out that yeast are interesting because they come in a couple of different mating types. It's essentially like sexes of yeast. And in order for them to mate with each other and go through a round of sexual reproduction, they have to be able to contact each other. They aren't motile, so they don't have the ability to move towards each other, so what they do is they grow towards each other. And in order to know which direction to grow, they each release these peptide mating chemicals. They're called pheromones. One yeast can sense the other yeast pheromone, and then it responds by growing in the direction of that other yeast cell. They fuse, go through a round of meiosis, generate a bunch of different new sexually formed yeast cells. That might not sound very interesting because it's yeast, but it turns out that that same concept of using chemical signaling and gradients is also important for development in multicellular organisms. So when you look at the development of neurons and the vasculature, those are situations where a cell has to figure out which direction it wants to grow an axon so that it knows how to get an axon from your brain down to your toe. Or for the vasculature, right? To make sure that you have veins and arteries in the right locations. They have to have signals that let them know how to grow in the right directions. And a lot of those signal transduction pathways that the yeast use are very homologous to what you see in humans, to the point where you can replace the human gene for some of those with, or you can take the human gene and throw it in the yeast, and the yeast will use the human protein just like it uses the yeast protein. That's how similar they are. That was probably about a minute and a half elevator pitch for work that I did like 15 years ago. But whenever I would show up at a yeast conference, I could throw out my elevator pitch, right? And people would know what I was talking about. I could also throw that same elevator pitch out at Society for Neuroscience. I went to that conference because axons, that's in the brain, that's neuroscience, right? Those two fields are related. And now the neuroscience person knows, oh, okay, this is a yeast researcher. Okay, I can make a few tweaks and modifications and actually use that same type of pitch in a couple of different settings. And it doesn't matter whether I'm talking to one person or 10 people, or if we're on the escalator going to and from different parts of the conference, or if we're on a random shuttle going back and forth. I can use my elevator pitch to start a conversation with another person without having to worry about what am I going to say, what am I going to say, right? So it's a really useful tool. Again, why use that elevator pitch? One, it helps people remember who you are and what you do, right? That's why when I asked you guys to tell me about your question, I wanted you to start with who you are and where you're from, right? If somebody hears a really great pitch and they're thinking, oh, that might be somebody I want to collaborate with, but they can't remember who they are or what institution they came from, and it's a conference that has a couple of hundred people or maybe 10,000 people, they won't be able to contact you later on, right? So you want to, have that memorable piece. Again, it helps people put you in the field. We'll talk in a couple of slides about modifications that you can make to your elevator pitch, but essentially, if I go to like a big conference like that neuroscience conference, it lets people know that, okay, you're in basic research, you're not in clinical research, and you're interested in a model system that we could use to learn more about neurons. Okay, gotcha. Now I know how to provide feedback or what types of questions I can ask and expect you to have an answer to. Right? Back then as a grad student, if I gave that pitch and then someone responded with, so what do you think about hippocampal synaptic formation? I would go, I work on yeast. They don't have brains and hippocampi. Right? So it's useful for the person that you're giving that pitch to kind of understand where you are relative to their research and whatever things are going on around them. And again, it opens the doors to future conversations and opportunities. Part of that yeast pathway involves a G protein coupled receptor. If you had to take biology classes like your 200 level sequences or even a few other, like maybe some psychology, lots of other places, G coupled protein receptors are all over the place. So it gives people a chance to go, oh, well, I'm interested in that thing too. And even though you're in yeast and I'm working on, I'm looking at the retina in humans, there might be things that we can learn about each other's G protein coupled receptor work that I might be able to share. Oh, hey, I'm a yeast researcher too, but I'm not interested in this. I'm interested in this other part of that pathway. Maybe we can work together, right? There's a lot of collaboration in science, and a lot of that starts with conversations that people have. And that's why we make you think about networking, right? These things are kind of linked together. So, that elevator pitch lets the person know what you're interested in, and it gives them the ability to maybe think about how their work might touch your work, and that can lead to some great things. If you've ever looked at the average scientific journal, it usually doesn't have one person. It's usually got groups of people, and those larger articles are usually collaborations between a number of different labs, maybe even on different sides of the planet. Okay, what's in an elevator pitch? All right, things that I put into my elevator pitch. I actually started with like my name again because that's good information to have. Topic, right? My topic right at the very beginning is I'm interested in yeast mating. So now the person I'm talking to knows, oh, okay, they care about how yeast can have sex, go through sexual reproduction. Okay, gotcha. I know what you're talking about. What's the problem? Okay, the problem is that we don't totally know how they establish these gradients. Okay, cool. Now they know that. Why is this interesting? If you don't know anything about yeast, you're probably going beer and bread. That's why it's interesting, but then I make that connection to axonal guidance and development of vascular systems so that people can then go, oh, okay, yeah, I get why that's worth maybe throwing some money at. Okay, cool. And then how does your work fit into the broader discipline and what does it add? Those two things can sometimes be linked together, right? The more you understand that model organism, the more you may be able to use that knowledge to apply it to other more complex systems. Right, so that's just an example using my particular piece, or pitch, how you can work through these 4 areas. Now we get to some of the fun part. How do you actually craft that pitch? Now remember, the banana and the orange, right? Usually the first kind of obstacle, hurdle that you have to overcome is just figuring out what kind of question you want to ask, right? And from there, you can sort of start framing your elevator pitch. I don't want you to get super stressed out about that first hurdle. I've got a slide coming that's going to talk about why I don't want you to get super stressed out. But the first piece is just kind of like figuring out what types of questions you're interested in, right? So your elevator pitch should at some point identify who you are, get into what your topic is, and your topic might again be really broad, right? Your topic for our activity was, I'm interested in oranges and bananas. A real elevator pitch, you might be interested in health disparities in Guam. You might be interested in depression. You might be interested in diabetes. You might be interested in access to clean water, right? Those are all pretty broad topics. And from there, hopefully you can find some more specific question that you're trying to ask. I'm interested in access to clean water in the Salem area, because if you're local, then you know that we've had some trouble with clean water in Salem. You might be interested in diabetes interventions for inner city children ages 12 to 18, because there's lots of issues with interventions for that demographic, that age group, right? You might be able to kind of narrow things down. And then there's always the so what question, right? Like, why does anybody care? Why does anybody care that I'm working on yeast? Oh, because it's useful for humans, and you're a human. And wouldn't you like, during development, your brain to function properly? And if we are going to do things like address neurodegenerative disorders with stem cell treatments. Don't you want those new neuronal stem cells to form neurons and axons that go the right direction? Right? You want to kind of think about that as you're developing your pitch. That's sort of that so what. The other thing that I like to add is that you want to make sure that you've got some hooks, right? Things that are in there that are going to activate the listener, right? Things that are catchy. That can be really helpful with a hook. And try to insert a little bit of your personality in your hook, right? You don't have to be completely deadpan. You can use your personality to drive that elevator pitch. And in fact, it's a lot better when you have that little bit of your personality driving that pitch. How many of you have taken a class where for the entire time your instructor was pretty monotone and boring? And were those your favorite classes to take? Yes, I know. There are a couple of faculty that we have at Clackamas that I've heard students tell me that the class was super hard and there was so much homework, but the instructor was really engaging. So I didn't really mind doing all the work because that instructor was really into it, and so it made me kind of willing to do the work, right? You're basically in that same role when you're presenting your elevator pitch. If I was to stand here and go, so I'm interested in signal transduction pathways in yeast and like how they talk to each other because they use these like funky peptide things to talk to each other, you would probably immediately go, oh, well, that sounds great. Right? You would wander away. But when somebody shows that they're enthusiastic and really interested in their project, it tends to make the other person listening interested and enthusiastic about that project. And if you think about it in that networking lens, right, you want to be able to make those connections. If the person you're trying to make a connection with walks off, that's a connection that's lost, right? But if they hang out for a few minutes because you're engaging during that elevator pitch, they hang out and they go, oh, okay, yeah, this is interesting. And they might not even have something in their lab that they're specifically able to connect you to, but they might be going, oh, there's a guy like third floor PSU that does yeast research. I wonder, like, maybe these two could talk and maybe there's something interesting that they could do together in collaboration. And they go share your story with somebody else because you had that extra enthusiasm, extra bit of personality, and that helped that person really remember your information, right? Helps you to get that second node away in your network, right? So that is another good thing to try to insert into your elevator pitch. A lot of times people, and I think if you have your little, does everyone have the elevator pitch guidance sheet? You should have that in front of you. I think they handed it out while you were thinking about oranges and bananas. Couple missing. If you look at that second bullet point, it talks about things from your lived experience, prompts you, or what from your lived experience prompts you to imagine the research, academic pursuit you are exploring. Does it connect to family members, neighbor, teacher, course, owns the exhibit, book, movie, something in your community? Right, those are great ways to insert a little bit of your personality. Don't, at the same time, feel obligated to feel like you're forced to put in those things. Like if you have a great story, like you're interested in maybe cancer research, and you're interested in that because your uncle and your grandfather both died of colon cancer at the age of 40 to 45, and so that's something you're really passionate about. If you want to share your story that way, that's great. If you just want to focus on, I'm really interested in colon cancer, that's also fine, right? Share what you feel comfortable with. As you grow as an individual and as a researcher, you'll probably figure out that there are other things that you're more comfortable with sharing. Right now, this is all new to everybody, so don't feel like you have to dump in all that information. Sometimes we have students that are interested in suicide because their brother or sister or someone else in their family unfortunately went through that, and so they don't necessarily want to share that story, and they're not ready to share that story. So don't feel like you have to share that story, but that can become a great way to connect people to your research and can also help give you that extra bump of enthusiasm. The second project I worked on as a postdoctoral fellow was related to Fragile X syndrome, and that one was fun for me because I have a nephew and a brother-in-law that both are in the autism spectrum. And so learning how this particular protein moves RNAs out into synapses and regulates translation in the synapse was my way of being able to use my molecular biology tool set to answer questions that were important for my family. Right, and I'm totally fine with sharing that information. When my nephew was born, I wasn't totally ready to share that information. It was kind of tricky to work through as a family. So now I can do that without any trouble. So again, feel free to insert things that you're comfortable with sharing, but don't feel obligated to just unload everything if you're not quite ready to unpack all of that quite yet. I told you this slide was coming. Elevator pitches change. They change for a lot of reasons. Some of it you're going to experience probably over the course of the next year. Because a lot of people, well, not a lot of people, all of you are going to develop an elevator pitch by Sunday. Don't worry, you'll be okay. Probably a year from now, you'll look back and you'll go, oh, actually, I kind of want to do this. Or you'll go, oh, this was a great base, and now what I'm even more interested in is in this one little piece of this elevator pitch. Or I had no idea what I was doing last year, and now I totally know what I'm interested in doing. Right? So your pitch is going to change. On top of that, as your progress from an undergraduate student to a graduate student to maybe a postdoctoral fellow to maybe junior faculty and then the actual person running the lab like Carlos and Lorena who was talking yesterday, right? As you get to those different levels, the access you have to different questions changes. So for me, some examples were, like I said, I went from the yeast lab and then I transitioned to another lab that studied Fragile X syndrome. From there, I went to a lab that studied honeybees. And we actually looked at the transition from nurse stage to forager stage, because it turns out that there's some differences in fat deposits. And those that fat deposit distribution is probably impacted by changes in gene expression. So I looked at proteins that are DNA binding proteins that are related to gene expression at the epigenetic level. So I looked at these histone binding proteins. And it turns out, so I looked at yeast, and then I looked at tissue culture cells for Fragile X syndrome, and then I looked at honeybees. Those seem like really crazy, unrelated things. But it turns out at the end of the day, what I was interested in were proteins that bind nucleic acids, how those proteins change their distribution patterns in response to external stimuli. Right? That really ended up being my big umbrella question, and so all of those projects were different ways that I could kind of understand how those processes worked. I'm going to tell you a secret. When I was an undergraduate student, nobody told me about elevator pitches. And when I was a graduate student, nobody told me about elevator pitches. And you just sort of develop those things because you figured out, I'm going to go to this conference, what do I say? And it was probably a year into my first postdoc that I said, oh, I see what the theme and my interests are. Cool. So you guys are officially about 8 years ahead of where I was when I started doing this. Again, thank you, Build EXITO. So that's one way where elevator pitches change, right? As you change projects. The other way they change is based on your audience. Okay, answer honestly, I don't mind. Raise your hand if many of those jargony sounding science words were words you've never heard before. Again, I don't mind. Okay, yeah, right? So if I'm giving some of those pitches at yeast symposiums, I would actually bump up the level, right? I would start talking about fuse 3-MAP kinases and sterol 12 scaffolding proteins and all the other things that go into the yeast pathway. If I'm at a Fragile X symposium, I would start talking about Fragile X isoform 13 versus 21 versus 27 because there's a whole bunch of different forms, right? I would go upwards. If I'm at a different type of setting, I would go down. So my best example is I would show up, you guys might not believe this, but it's true, we do get time to do things as researchers. I would show up at parties as a graduate student and want to talk to other people about like what I'm doing in the lab. And I'd be like, yeah! So I'm studying this really cool G-protein coupled receptor. It's got these 2 subunits, and the G-alpha subunit, we've got a version that has these really cool like temperature-sensitive mutations. And so if you change the growth parameters, then it activates the G-protein, except it's not supposed to be activated. So then the yeast would like basically respond to pheromone when there's no pheromone around. And my friends would be like, oh, yeast. Whoa. Right? Wrong audience. So instead, after many failed party attempts, I realized that basically what I was talking about was yeast having sex. So I would go, yeah, so I spend about 5 hours a day watching yeast have sex under a microscope. And they would go, oh, that's cool, how's that work? And then I would go, okay, so you know at the Museum of Science Industry in Chicago, they have those cool mice that like glow green under the UV light? Yeah, I've seen those mice. Like, yeah, so I take that protein, I put it in the cell, and then I can see where the colors are. And they go, oh, that's really cool. And I go, yeah, it is really cool. And they don't walk away. Right? So I change it in order to make sure that I'm interacting with the individuals in the audience at a level that they can understand. My second fun example is I went to this really cool, I was in a program that was similar to this, kind of midway through graduate school. It's called this Illinois, what is it? It's the Illinois Minority Incentive Program, and it was designed to increase the number of minorities that would go on to academic positions in the state. I did it for a while, but this is Oregon, I know. So we would have these big conferences, and it was a very similar, like very wide breadth of researchers and students that would come to this conference. And I gave my yeast poster, like presented my poster, and I'm thinking, yeah, there's like science people, so I'm just going to aim for my usual science level. I'm good. G-protein coupled receptors and pheromones, I'm good. And a group walked up to me, and I went through my talk, and at the end, one of the students says, oh, pheromones. Okay, so like you study pheromones that like humans make? And I went, yeast? And pheromones in humans? Like that, I don't know where that's at right now. I mean, this is probably about a dozen years ago. And we ended up in this wonderful side culvert that I had no idea what they were talking about, because they were coming from more of a like behavioral science aspect. And the reason that occurred was because I didn't have my pitch set properly for that audience, right? If I had included a few more pieces and tweaked it a little bit, then I wouldn't have ended up going down the side pathway that made me seem like a crazy person that didn't want to interact with them, because every question that they would ask, I would go, I don't really know, and that's not really where my research focus is. Right? And if you're thinking about that, hey, this is a way to develop networks and find people that can connect to your research. If your responses are all, that's not really me, that's not really me, that's not really me, then you get, okay. Right? So again, making sure that you're kind of targeting your audience really helps. The pitch becomes natural over time for a few reasons. One, because you'll probably give a pitch of some sort a lot. Right now, it's going to seem kind of, not necessarily artificial, but sort of forced because we have this as an activity because it's a thing that you want to learn how to do. So it's going to feel a little uncomfortable. How many people have taken a college class before? And how many people were uncomfortable for maybe 3/4 of the class? That's because you were learning something, right? I can remember as a kid learning how to ride a bike, and it was uncomfortable for many, many weeks because I was learning. And then once I learned how to ride a bike, I could hop on the bike and I could do whatever I wanted to do. And once I got a little bit more practice, I could pop a wheelie and I could jump over curbs and I could roll downstairs, right? I could use my bike in all sorts of other situations. I could, ride on the back pegs, I could pedal somebody with me, right? But when you're learning, none of that stuff even sounds feasible, better yet comfortable, right? And then that's what this process is gonna be like. So it's okay for it to be a little bit uncomfortable. But once you get comfortable in it, you won't have any trouble making those adjustments. It just becomes more of the thinking in advance. Okay, where am I going? Who am I talking to? What's their background? What's the setting? Am I presenting my work in a K through 8 classroom because it's an outreach thing? Okay, I need to get to the K through 8 level. Am I doing it as part of a larger undergraduate symposium at PCC? Okay, I need to be aiming for that level, right? You'll be able to identify those things and adjust as needed. The other thing is that this elevator pitch, even though it's an oral presentation, it really is just an example of how to be concise with your logic and thinking in science. And that is really a huge, huge, huge thing that you want to start developing over time. Raise your hand if you've ever seen a research article, like a piece of primary literature, some journal article someplace. Okay, so that's a lot of people. Do they all start mostly with an abstract? And that abstract is maybe 200 to 350, 400 words. That's a really concise and brief story of what's in that paper. And if you read it to somebody, it would probably take you about a minute to a minute and a half to read it. It's an elevator pitch, right? It's an elevator pitch for that article. How many people have heard people that have come up here talk about grants? Right? Part of the thing that you have to do as your grant is you basically need a little abstract that basically gives the grant reviewer, who's going to decide how much money you get, right? They want to have something that they can look at and in a really short period of time go, yeah, this seems great, or what is this, right? That's a sorting mechanism, right? So if you can learn how to get concise and have a nice logical flow that people can read and understand, then it's going to help you get journal articles written and submitted and accepted. It'll help you get grants written, submitted, and accepted, and we want to see lots of people get their grants, right? It helps you in all of those different areas, right? That whole idea of being concise. So, it's a good way to practice that skill. Once you get it down orally, it will translate into the written versions of these elevator pitches. Even the thing that you guys did yesterday, where you talked about your sites, that was an elevator pitch, right? You gave an elevator pitch for your site. We gave you more time, because there's a lot of you, but it's an elevator pitch for your site. Some tips on ways to help develop your question. First, again, think about things you're interested in, right? Are there things in your community? Are there things from a class that you took? Are there things that maybe you just managed to hear on, okay, I'm gonna sound old, hear on NPR, maybe this morning on your ride in, heard on the news, heard on Facebook or Instagram? Please fact check, right? Things that interest you. In some cases, depending on the topic that you're starting from, you might have to broaden things out or you might have to narrow things in. If you have a very, 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 very specific thing, like the, diabetes in 15 and 18-year-olds in an urban setting, because that's where you grew up and a lot of your friends suffer from diabetes. In your elevator pitch, you might have to spend a little bit of time backing out, right, and talking about diabetes and the cost of medical treatment for diabetes. Like, you might have to broaden it before you get to your your initial thought and question. In other cases, people have a really broad idea of what they're interested in, and then they have to take a little bit of time to try to narrow it down to something that the listener will be able to connect to. Do some research. And again, not Facebook, Instagram-based research. But you can take, raise your hand if you have a phone with access to the internet. Yeah, so most of you are plugged in right now with computers that are better than the computers that I used when I was your age, like in your pocket. And I'm not that old, right? So you guys all have access to lots and lots and lots of information. Now, don't go into information overload, right? It's really easy to pull up the most recent 4 reviews on breast cancer. You're not probably going to be able to synthesize that down into 1 minute and introduce yourself and explain why it's important to you, right? So you'll have to kind of cherry pick. I would say to try to focus on things that seem like they would be impactful to your listener, right? Think about those things. If you tell me that there's a high incidence of diabetes in urban areas, I'm like, oh, that's kind of cool. If you tell me that there's a 7 times increase in diabetes in urban areas, yet they receive 20% of the medical care versus other areas, then I go, wow, that's crazy. Please fact check. I just made those numbers up. Don't use them in your elevator pitch. But right, like that extra bit of information can really go a long way with keeping that listener involved and engaged with your elevator pitch. And then practice. We will have lots and lots and lots of opportunities to practice throughout the next, 3 days. We will give the elevator pitches on Sunday, and they need to be about 1 minute, 60 seconds. It's not very long, right? If you're not careful, it can take you 60 seconds to introduce yourselves. So, you don't really get a lot of time, and we will actively enforce the 60-second time window. If everyone gives a 5-minute elevator pitch, most of you will miss your flights back. Right? That'll take us 10 hours. So we really do have to keep it down to a minute. 1, for logistics, and 2, because if you start going on and on and on and on and on. So I look at yeast, and they have like these really cool cell walls, and they kind of they look like this, and then they go down, and like you can stain them, and they make pretty colors, like you're like, oh god. Right? You want it to be concise enough that the listener also doesn't become disengaged because you've added so many unimportant things that they don't know where you're going. Right? So that 1 minute is really important in a couple of ways. All right. Now, if technology works with me, who wants to see a few examples of some elevator pitches? Okay.
Hi everyone, I'm Brandy Lentz. I'm from Tory Community College. I'm working on my undergrad degree there. I'm going to go to PSU and then get a bachelor's of public health and then to a master's of epidemiology. My interest area is severe psychological trauma and how that affects the immune system. More specifically, people with severe trauma have adverse health outcomes in general, but most specifically when it comes to autoimmune diseases. The problem that I see is that oftentimes people who have severe trauma are diagnosed for mental illness, and their physical symptoms are sometimes attributed to that mental illness, and then the diagnosis for the diseases that they have can take years to happen, and after that point, organs can be damaged, and these people have been living in pain for a long time.
So, did we hear name and where she came from? Okay, we got that. Does everybody have a pretty decent idea of what she's interested in? Okay, did we have why? Like why that is an important thing to understand, right? Those were in there. It was about the right time. I think it might have been maybe 10 seconds-ish over. But it's got those key things. This student was a student that was in this same room a year ago, starting off from the same position. Now, nerves and public speaking backgrounds and things like that can make things different. But this is what she was able to come up with during the course of our 4 days. So again, that's why I say you'll be fine. You will be able to tackle this obstacle once you kind of like get an idea of what you're trying to do, what you're aiming for, and what you need to have in that pitch. You'll be okay. Don't worry. And there are lots of people. There's not quite 100 of your peers. There are probably 20-ish faculty. And I would say in the grand scheme of what's a safe place for me to practice delivering an elevator pitch, this is the safest place that you will find to be able to practice with this many people ever. Right? You're not probably going to be able to walk into a room with 120-ish people that are going to have zero judgment and only have good intentions for what you're trying, like the work you're trying to do, and only want to help you get better. Right? Some of those other settings, there's either going to be judgment that's involved because they're trying to decide, do I want to sit and listen to this, or do I want to move on? There's going to be money involved because they're looking at your abstract for your grant and they're going, do we want to give this person $50,000 or do we want to give this person $4.5 million over 5 years? Right? There's that, or there is the publication component that they're thinking about. Do we want to publish this person's thing or this person's thing? Right? None of that exists in this room during the course of these next 3 or 4 days. Right? So take full advantage. And I'm pretty sure, I'm probably going to throw some faculty under the bus, but I'm pretty sure you can walk up to just about any faculty member from any of the colleges or universities and say, hey, would you be willing to listen to my elevator pitch for 2 or 3 minutes? And they will listen. Every year I listen to like 30 random elevator pitches, 5 of which are Clackamas students, and the other 25 are from wherever. Because all of the faculty here really want to make sure that you have that ability to get feedback and learn how to develop this wonderful process, right? You won't master it today, but at least if you can get your feet wet and feel comfortable doing it, it'll help you going forward. So, it's a great place to learn. Okay.
Hello, I'm Kason. I'm a student here at Portland State University, studying mechanical engineering and video microbiology. My main focus for research is my interest in the indigenous people. The main prevalence of them is in Papua New Guinea and the Dominican Republic and islands in Asia. And the indigenous people have what is known as 5A reductase deficiency. And 5A reductase is an enzyme that typically is produced in about 8 weeks. When an XY fetus, the brain starts releasing the enzyme that tells testosterone to convert into hydrotestosterone, which is what causes the peripheral tissue like your skin to convert from female genitalia to male genitalia, and what we would then call cisgender male. This does not happen with them though, and so when they are born, they have an XY chromosome and a male gender identity, but have female genitalia. When they hit puberty at 12, testosterone is introduced into the system, the enzyme is activated, and testosterone starts being converted to hydrotestosterone, which at that point the female genitalia starts converting into male genitalia and sperm production begins. They then develop into normal cisgender humans or functional adults and live normal lives. My hope is to research that process and apply that to transgender individuals in hopes for specifically transmasculine folks and improving their surgical options with the eventual goal of reducing the suicide rate, which is around 46% in trans individuals, close to the national average, which is about 4%.
I wanted to include that talk because the front half seemed really like bulky science heavy. There's lots of testosterone and dihydrotestosterone, lots of like words, physiology, science, that if you're not a biology or anatomy, physiology, medical person, might turn you off. But then the back half of his talk is really engaging. And the why and what, like why he's interested in that and what he hopes to learn and how he hopes to apply it, is really interesting and developed or presented in a way that I think most people would be able to connect to. So, if you are kind of going down a very like, lots of times it ends up being kind of the people interested in biochemistry, pharmaceutical, those types of areas, they can be a little bit complex. So you might have a little bit of complexity in your elevator pitch, but if you can turn some of that into language that most people can understand, then you'll still be able to get your point across and engage with the listener without the listener going glassy-eyed and wandering away. That's also where being concise helps. Like imagine if he gave you 20 minutes of that piece that then led to the 1 minute of, I would like to use that to improve the health disparities with transgender individuals and how that transition works. Right, you would have wandered away probably 5 minutes into that without getting his really good hook at the end. So that's where being concise can really help.
Hello, my name is Mackenzie Pegracian, and I'm currently studying psychology at PSU. In high school, I was the president of the Gay Straight Alliance for 3 years. In this position I held, it generally made others comfortable coming to me for education and emotional support when it came to LGBTQ issues. What I noticed over the time I served is that a majority of those I worked with struggled with mental illnesses such as depression, anxiety, and PTSD because of bullying and home abuse. I did further research and found startling statistics. LGBTQ individuals are 3 times more likely to experience a mental health condition and 4 times more likely to engage in self-harm or attempt suicide. This statistic can go up to 8 times when they lack family support. And on top of all of this, there are still disparities in healthcare because of stigma, prejudice, and a lack of overall education. Because of this, I'm interested in studying the effects of trauma and maltreatment on youth in this community and how this affects brain development, emotional and behavioral functioning, and overall mental health.
Okay, so that was almost exactly a minute. I think she went like 1 minute and 6 seconds or so. But you can see all the elements that we've talked about, right? She had done some research, she had a few key numbers in there, but not really overwhelming or anything. Notice she had her notebook. You are not the first cohort to get really cool build EXITO notebooks. So it's okay to have some notes and bullet points to refer to, that's fine. If you have a great memorized, like the last 2, they had their pitches pretty much memorized, and that's fine. But if you need something to help you kind of make your way through your pitch, that's also fine. I have notes up here. It's okay.
Hello. Aloha. Talofa. Buenas and Afane for Samsung. I come from the beautiful island of Roller. It's one of the 3 islands in the cinema. And I am currently a student at the Northern Rivers College. Did you know that diabetes is one of the leading causes of death in the cinema? In addition, we are considered 3rd in the world for diabetes based on population. So in the cinema, 83% of people with diabetes are indigenous, and out of the 82, and out of the 82%, 58% of them are women. So for my research, I'm interested in looking into the aspects within the Chamorro culture to help identify ways to decrease the rates of people developing diabetes, and also to gain a better understanding as to why this disease occurs more with females versus males. Thank you. This is Masi.
right, so one more example. And there's a really nice one. I feel like I could probably listen to him tell me about all sorts of cool things just because he's got a personality that just keeps you engaged. So again, like, use your personality to your advantage, right? If you can include some of that in your elevator pitch, it'll often help make connections between you and your audience. So feel free to insert those things. If you are interested in more examples of elevator pitches, you can do a YouTube search. We have the elevator pitch session from last year in YouTube. It's about 2 hours long, so don't go down a really long rabbit hole, but you can go to that rabbit hole. It might give you some inspiration, some ideas, and help get some of the nervous jitters out. So that part's okay. Speaking of nervous jitters, that's also okay, and that's going to happen, so don't worry about being nervous when you come up to present. I always tell this story. When I was in the Army, I jumped out of airplanes, and I would always see these people that were just like, they were not nervous at all, they were just ready to go. And I thought, oh wow, I must be doing this wrong. These guys are just like ready to jump out of a plane. This is crazy. And I had like this master sergeant who made like 500 jumps. He walks over to me because I look a little nervous, and I explained to him, I'm really nervous and nobody else seems to be nervous. And he goes, good, because those people are crazy. You're supposed to be nervous. You're jumping out of a plane. Okay, so public speaking, for most of us, will get nervous, and what usually will make most of us nervous. And usually what it boils down to is sort of coming up with ways to get over that nervousness. One easy way is to just be more familiar with everybody in the room, right? Have you ever had trouble talking to your friend about what you're doing today? Probably not, right? So spend some time getting to know the other people in the room, and that way you'll be able to make connections, eye contact with friendly faces in the room because you recognize them. You only need to know probably about 10 or 15 people to be able to make eye contact with the whole room. So that's one way to help get over the nervousness. I know that's something that students have trouble with during these elevator pitches. Again, 60 seconds is not very long. Focus on those big things: who you are, what you're interested in, why you're interested in it, because that'll help you make that personal connection, and then why the person listening to it should be interested in it, and what's the big thing that you're hoping you'll be able to learn if you can get that question answered. And if you can do that in a way that's engaging, then that's going to help that person say, ooh, let me help you answer that question. I have a friend. I have lab materials. I know this method that you might not be aware of that will help you answer this question. I am the one that is in charge of awarding all of the grants. I am the one that's that's the editor for Nature and has to decide which one of these articles is going to get published, right? It helps them make those connections. And once you get those connections, you can go a long, long way. Okay. With that being said, I think we've got about 15-ish minutes, maybe 20 minutes until our break. So what I want you to do with that remaining time is think about that big picture, concept, question, idea, right? Think about what some ideas for an elevator pitch might be. You've got a notebook, so you can take lots of notes, right? Brainstorm. Think about what some good questions might be. Use your group at your table to help you. And then after that, I think we have a break scheduled. So I'll stop you guys when it's time for your break.